Alright, this virtual lecture is going to deal with the prokaryotic cell wall. Now, if you're going to learn anything this semester, and I hope you learn a lot, I really do, I think this is a good course, but if you're going to learn one thing this semester, learn the structure and function of the prokaryotic cell wall. You're going to see this information is going to be helpful in chapter 3, 6, 7, 11, 14, 15, chapter 20. I mean, this topic is threaded throughout the course. So pay special attention. What is the function of the cell wall? What does it look like? What are the different types of cell walls we find in prokaryotes? So on this slide, let's first start with the function. What the prokaryotic cell wall does is it provides structure and protects the cell from osmotic lysis. Here's what that means. I'll give you an example of a human cell, which is a eukaryotic cell. Let's start with a red blood cell. Now, you may have learned about this in anatomy and physiology. As water rushes into a red blood cell, that red blood cell is going to expand in size, right? The volume is going to increase. Now, if water continues to rush in unregulated and the cell cannot adapt or the body can't adapt, what eventually will happen is that red blood cell will lyse open, it will burst like a water balloon, and all the inner contents will spill out. Now, that's a dead cell. Not good. Not good for your cell to burst and have the inner contents spill out. Now, for a prokaryotic perspective, we don't have this concern because around its cell is this rigid cell wall. So let's imagine a scenario now where water is rushing into the bacterial cell. Now, that bacterial cell is still going to expand. What eventually is going to happen is it's going to push up against the four walls here, if you will. It's going to push up against the cell wall. And what the cell wall is going to do is it's going to prevent further expansion. So that cell cannot get any bigger. Essentially what that does then is it prevents it from lysing open, or another way to think about that, it prevents it from dying. So it's a very important structural feature for a bacterial cell. Now, it has other indirect functions. In some cases, it can help cells attach to each other to form communities. It also is very useful in filtering antimicrobial drugs. We'll deal with that in a little bit. There are certain types of bacteria which have a barrier based on the structure of their cell wall, so the antibiotic can't get in and do its job. So we have to be very cognizant when you prescribe antibiotics. What is the infection you're dealing with? Because in some cases that antibiotic may be rendered useless based on the structure of that cell wall. Now, conversely, we can also use the cell wall as a target for antibiotics. And the answer to why is actually drawn on our slide. As we mentioned before, human cells lack a cell wall, whereas bacterial cells have a cell wall. So if you can imagine this, if you can target the cell wall for destruction, if you can destroy it, so no longer does that bacterial cell have a cell wall, now the simple act of water rushing in will cause that cell to die. So we can often target then the cell wall using antibiotics, which is a very important clinical feature of the cell wall. So let's talk a little bit now about what the cell wall is made of. A cell wall is made of a mixture of sugar and protein. Another word for that, it's made of peptidoglycan. That's an extremely important term that I would expect that you use throughout the semester. Peptido means protein, glycan means sugar. Now, when we look at the sugar part of peptidoglycan, what we're going to see is we have these two sugars called NAG and NAM that are threaded together to form layers, and those layers are then stabilized by what we call tetrapeptide cross bridges. That's the protein part of the bacterial cell wall. These bridges provide strength. They help to stabilize the cell wall. Let me show you a picture of a drawing of what the cell wall may look like. Now, first, here are those sugars that we talked about. Here's NAG and NAM. They're derivatives of glucose. That means we've modified glucose. You can see here at this position, we've modified them. Also here at the a third carbon. We've modified them as well, specifically in the NAM subunit. And so what we do is we take these disaccharides, if we're a bacterial cell, and we have these repeating units where we link them together. And that bond there, and that bond there, that bond there provides strength. So the sugar part has some inherent strength to it based on its bonding. But if we want to add to the stability, this is where the protein parts come in. So here we can see again, here's our repeating NAG and NAM. Notice here, not only do we have many strips of NAG and NAM all along the same plane here, we also begin to stack them together. And in order to stabilize these layers of NAG and NAM, we use here 4 amino acids forming this cross bridge. And we also have a series of amino acids here that also stabilize as we go across. So when you take into account the bonds in the sugar, and now the bonds as a result of those proteins, what we have is a very stable structure that prevents overexpansion. So now, are all bacterial cell walls created equally? The answer is simply no. There are 2 types of bacterial cell walls you'll be responsible throughout the semester. The first is called a gram-positive cell wall. Now let me talk a little bit about that name. There's a special staining technique called a gram stain, which actually refers to the scientists that devised the staining technique. And what they mean by a gram-positive cell wall is that when you use this staining technique, it provides a positive result. We're going to learn, and I want you to take note now, that when I stain a bacteria that has a gram-positive cell wall using this specialized technique, it's going to appear purple under a microscope. And purple is an indication of a positive result in the gram stain technique. So that's very important. So now when I start to think about gram-positive cell walls, I know they're going to stain purple in the laboratory. Now what makes a gram-positive cell wall unique? Well, we have a very thick layer of peptidoglycan. So if I were to go back a slide, this artist rendition that we see here, this is actually what a gram-positive cell wall would look like. You can see the many layers of NAG and NAM sitting on top of one another. So when I compare the 2 types of cell walls, gram-positives tend to have more strength than a gram-negative cell wall. So what are some of the other features of a gram-positive cell wall that makes it unique? They have alcohols called teichoic acids. These teichoic acids, for example, can provide antigenic variation. Here's what that means. An antigen is what's recognized as a marker. So different gram-positive bacteria are recognized differently based on the presence of different teichoic acids on their surface. It also helps to anchor the peptidoglycan to the cell membrane. Now, if you recall, a cell membrane acts as a barrier that controls passage both into and out of the cell. And so what you can imagine here, if this is a cell membrane, the cell wall surrounds it. And so what allows a gram-positive cell wall to remain anchored to the cell membrane, teichoic acids do, specifically lipoteichoic acids. There are also a very unique class of gram-positive bacteria called acid-fast bacteria, who have specialized acids called mycolic acid. These mycolic acids are very waxy, as we'll talk about in chapter 3. If you think what waxes do, waxes are impermeable to water. So what this results in is a bacteria that doesn't dry out easily. I'll give you an example of why that's important. Mycobacterium tuberculosis. Mycobacterium is the genus name. Tuberculosis, the species name. That bacteria causes tuberculosis, as you would imagine. Well, tuberculosis is unique in the fact that if someone sneezes or coughs, that bacteria can actually stay airborne for longer than most bacteria because it doesn't dry out in the air. See, the air is drier than the bacteria, so water is going to be sucked out. But for a bacteria with mycolic acid, it doesn't get dried out as easily. So this is a bacteria that can stay airborne for longer periods of time, which makes that particular bacterium very contagious. And so if someone's in the hospital with TB, you'll often see them quarantined. You have to wear masks when you're around them because of the idea that this bacteria can stay airborne for long periods of time. Here's a picture of what a gram-positive cell wall would look like. Also, review your textbook. They have a slightly different picture. I like this one because it really gives you the idea of the thickness of the peptidoglycan. The artist here has drawn it as bricks in a wall, the bricks being the NAG and NAM. You can also see the teichoic acids here. Here's an example of a lipoteichoic acid. Notice how it's anchoring it to this phospholipid bilayer called a cell membrane. Here's another lipoteichoic acid anchoring it here as well. So those are unique to gram-positive cell wall. So when I ask you, what makes a gram-positive cell wall unique? What are some of the things you're going to list? That's correct. The first thing you list, very thick peptidoglycan cell wall. The second thing you may want to list is the presence of teichoic acids. The third thing you may want to list is for a small subset like Mycobacterium, they have mycolic acid in the cell wall. Now let's move on to the second type of cell wall we'll see this semester. That's the gram-negative cell wall. Now the first thing I want to point out is this. Whereas a gram-positive cell wall had layers upon layers of peptidoglycan stacked on top of one another, here we have a very thin layer of peptidoglycan. Here it is in the picture. Notice the depth here does not nearly approach the thickness of the peptidoglycan in a gram-positive cell wall. That's the first thing I want to highlight. The second thing I want to highlight, which is going to be extremely important as we go through the semester, is the presence of a second membrane called an outer membrane. Now you're going to read in your lecture notes and in the textbook this semester, membranes act as filters. I just mentioned on the previous slide that the cell membrane controls what goes into and out of a cell. So here's the cell membrane here. If you want to orient yourself, here's the inside of the cell down below. Here's the environment up here. Now, when you look at a gram-negative cell wall, what we effectively have are two filters. We have this first outer membrane, which is going to filter what gets into this space called the periplasmic space. And once things get in the periplasmic space, now you have a second filter which controls what gets into the cytoplasm, the inside of the cell. So we're going to see gram-negative cell walls are much more difficult to treat with antibiotics than gram-positive cell walls because we have this extra barrier which is going to prevent things from getting into and out of the cell rather easily. So if I were to ask you on an exam, what is the most important difference that in a gram-negative cell wall compared to gram-positive? Certainly the thin layer of peptidoglycan is important to include, but for me, what becomes most important is that you identify the fact that we have an outer membrane that acts as a filter, which is going to prevent materials from easily getting in. So disinfectants, for example, which would clean the bacteria off of a surface, are not going to get in as easily. Antibiotics, which are drugs that are meant to destroy bacteria now within an individual, are not going to be as effective because they also get filtered out by this outer membrane. Here's another important difference that I want you to pay attention to. In this outer membrane, we have a substance called lipopolysaccharide. Lipo meaning fat, polysaccharide meaning sugar. Now this lipopolysaccharide is extremely important when we start to talk about why bacteria can cause illness. We often call this particular structure an endotoxin. Now that word toxin is ominous right there. Now when I look closely at lipopolysaccharide, the lipid part of it is called lipid A, and this is the business end of lipopolysaccharide. Lipid A is what gives it toxic properties. Here's a picture of it. You're never going to have to memorize this, folks, but here's what I want to point out. It's the lipid A that embeds itself into an outer membrane. Now here's what happens. When a cell dies, we talked about this before, essentially the cell lyses, everything spills out, and eventually even the membrane and the cell wall get degraded. Now, as that membrane and the cell wall gets degraded, lipid A gets released into the environment. Well, that environment may be your colon. That environment may be your bloodstream. And as lipid A gets released, it causes a systemic reaction. Now, locally, it's going to cause inflammation. So when you get food poisoning, what happens is, as lipid A gets released, it causes inflammation. That inflammation leads to cramping and a lot of the symptoms you associate with food poisoning. From a systemic point of view, if lipid A gets released into your bloodstream, it's going to cause your body temperature to rise. It's going to cause your blood vessels to dilate, which is going to allow their transport to occur at a much faster rate. It can cause shock. It can cause blood clotting. It can, in high levels, be deadly. You may have heard of septic shock before. Septic shock is not just high levels of bacteria in your blood. Septic shock could be to high levels of just lipid A in your blood, which can cause a whole host of cell responses. Now, why would this get released? Well, check this out. When we treat infections, they can cause lipid A to be released as the bacteria dies off. So if you think about this, and pardon my language, I hope I don't offend anyone, but this is a bacteria's way of saying one last F you as you kill it off. Even as it's dying, it is causing you illness. So for example, for food poisoning, we often don't get an antibiotic. What the doctor will tell you is just let it run its course. Because if I give high doses of antibiotic in a short period of time, all that lipid A is going to get released, and it's actually going to exacerbate your symptoms. It'll actually make it worse. So you're better off allowing your body to slowly clear out the infection, trying to keep levels of lipid A at a constant low level, which meaning that you'll have prolonged symptoms, but the symptoms won't be as severe, and eventually they'll trail off. So we've introduced the idea that we have this outer membrane. We've introduced the idea of lipopolysaccharide. We also have this periplasmic space. The periplasmic space is important because it can store nutrients, has water, other substances that's secreted by the cell. It's sort of the conduit between the inside of the cell and the environment. It even has digestive enzymes, so the bacteria can start the digestion process outside of the cytoplasm. So digestion can start here, breaking things into smaller materials, and now we can bring that in. One other structure I want to point out besides lipopolysaccharides and the outer membrane itself has a series of proteins called porins. Porins are transport proteins that are going to regulate what gets into and out of the periplasmic space. Now that's going to be important. For example, when a bacteria becomes resistant to antibiotics, one of the ways it can become resistant to that antibiotic is by changing the porins on its surface. So no longer will the antibiotic be able to get through if that gets changed. Therefore, the bacteria will no longer die in the presence of that antibiotic. That is the definition of an antibiotic-resistant bacteria. Alright, so now is a good time to test ourselves to see if we really understood the information that was presented. Now, as we talked about, selection of the proper antibiotic is critical to treatment of a bacterial infection. If you don't treat an infection with the right antibiotic, what you're really doing is allowing the bacteria to propagate. You're allowing it to take hold and cause more damage during that time when you're on a drug that's not working. So it's important that we pick the right drug. Now, some bacteria naturally are resistant to antibiotics. due to the structure of their cell wall. These are going to be more difficult to treat. So the question I have for you is this: will it be a bacteria that has a gram-positive cell wall that's going to be difficult to treat, that's going to be naturally resistant to antibiotics? Or would you select a gram-negative cell wall as the one that's going to be naturally resistant to antibiotics? So what I'd like you to do now is press pause and either use your cell phone or go to pull4.com and submit that code for your answer. So if you think it's gram-positive, submit 150853 to the text number above, or go to pull4.com and type 150853 in the box provided. All right, I assume everyone's done that. What is the correct answer? The correct answer is a gram-negative cell wall. A gram-negative cell wall, as we talked about, has an outer membrane that acts like a filter, and because of this filter, there are many antibiotics that can't traverse that layer, making the bacteria naturally resistant to many antibiotics. So the best answer here again is gram-negative. Here's actually an article I took online. I think I took this from CNN.com. I've sort of parsed it down, and I've highlighted those terms that I think we should understand now when reading an article. So here we have an example of an infection that was acquired in a hospital. We often call this a nosocomial infection. Here we have an athlete in his younger days, Richard Armbruster, played baseball into his 70s, which you can imagine is impressive. He ended up developing some pain in his right hip, and it turns out he needed a hip replacement. Now, while this is invasive surgery, they're fairly routine. However, by late March, Mr. Armbruster never made it out of the hospital. He unfortunately died. And the reason he died was not due to hip pain. The reason he died was from post-surgical complications, in which an infection was acquired, which spread to his bloodstream. And the problem for Mr. Armbruster was that the bacteria that he was infected with was resistant to the antibiotics they attempted to treat him with. And so by the time that they figured out the right antibiotic, it was most likely too late, and the toxins and the bacteria in his blood has risen to uncontrollable levels. Now, read this article, right? This is not from your textbook. This is from an article from CNN.com, from a website. The bacteria classified as gram-negative. Now, we know what gram-negatives are. Right, I'm going to ask you a question, pop up on the screen. Describe a gram-negative bacterium. That's right, gram-negative bacteriums have an outer membrane and a thin layer of peptidoglycan. So there are certain bacteria that are gram-negative that cause pneumonia, UTIs, infection of the bloodstream. And it says here their cell structure makes them more difficult to attack with antibiotics than gram-positive organisms, like methicillin-resistant Staph aureus. So can you tell me why their cell structure makes them more difficult to attack, naturally speaking? That's right, because they have that outer membrane that acts like a filter. So again, this is not from your textbook. This is everyday, an everyday article where now you can start to read and dissect it. Now check this out. Stats from Europe, where hospital surveys have been conducted. Gram-negative infections are estimated to account for 2/3 of 25,000 deaths each year caused by hospital-acquired infections. You hear all the time about MRSA, and certainly MRSA is a problem, but if you look, for example, in Europe, 2/3 of the hospital-acquired infections are not caused by MRSA. The deaths are caused by gram-negative infections. Now, why is a hospital a great place to get an infection? We'll talk about this later on, but of course, you have patients whose immune systems are already compromised. Here we have germs that can survive for long times on surfaces in the hospital. They can enter through surgical sites or wounds. They can enter through ventilators. Now, when we talk about the gram-negatives, it's not how frequent you find them, but the inability to really treat them. You see this quote. I think this is a great quote. For gram-positives, we need better drugs. For gram-negatives, we need any drugs. And this is from an infectious disease specialist from UCLA. I mean, this is a real concern, folks. And the reason is, restating it, is that gram-negatives have that outer membrane which acts like a filter. And because of that, many of the common everyday antibiotics are not effective, allowing these particular infections to take hold and in some cases, like Mr. Armbruster, cause death.